There are disclosures, of course. I'm one of the owners and a founder of Trinity Orthopedics, which manufactures the device. I'm one of the inventors. I use the device in clinical practice. I'm currently clinically retired, but I'm a paid consultant for Trinity Orthopedics. Ultimately, the objective of any bone grafting procedure is to um, produce bone at a distant site and ultimately for that bone to undergo incorporation and remodeling. But that process can occur with a variety of substances, both autologous and non-autologous bone, as well as bone graft stimulators. But we know that fusion rates are not identical. There's a variety of reasons for that, but the bone graft is likely one of the components for affecting the likelihood of success. The CoreX device is a sterilely packaged single patient use device. It does not require any assembly or disassembly during its use. It does not require any supplemental instrumentation. Now, some will contend that autologous cancellous bone is no longer the gold standard for a variety of reasons, but I would like to challenge that contention. We have greater than 100 years of peer-reviewed journal articles demonstrating the efficacy of autologous cancellous bone in both experimental and clinical applications. And in very few of these instances do the authors have any potential conflict of interest, which cannot be stated for many of the orthobiologics that are being touted as substitutes. Also, there's no other bone graft or bone induction material that's as rapidly incorporated into the host site. This is a quote from an article that was published in the Journal of the American Orthopedic Academy. Neovascularization occurs within the graft as early as 2 days after implantation, and it continues to have repopulation of the marrow spaces with both donor and recipient origin mesenchymal stem cells. Well, allografts are frequently used and they have their place, but they're not immunologically inert or privileged. Freeze drying or freezing does reduce allograft immunogenicity, but it does not eliminate it. Again, from a text called Bone and Cartilage Allograft, Chapter 1, I'm quoting here, bone allografts are influenced by the same immunologic factors as other tissues. The rejection of a bone allograft is histologically expressed by disruption of vessels as inflammatory processes, including lymphocytes, fibrous encapsulation, peripheral graft resorption, callus bridging, non-unions, and fatigue fractures. And this is because there are protein elements within the allograft which are immunogenic. Now, adding mesenchymal stem cells to allograft does not create an equivalence to an autograft. First of all, allogeneic bone grafts contain live mesenchymal stem cells that have been expanded in vitro. Now, it must be understood that these cells have to differentiate. They have to become mature cells and express proteins, and that this maturation process is heavily influenced by the microenvironment. Various cytokines and a very specific microenvironment have to encourage these mesenchymal stem cells to differentiate into the cell line that produces either cartilage or bone. And so just adding these expanded cells to allograft does not reproduce the microenvironment. It should also be understood that while it's often contended that mesenchymal stem cells are immunologically privileged, this is inaccurate. Both cellular and hormonal immunogenicity occurs in vivo. In vitro, that's in the test tube or on cell culture, there is some immunologic blunting, but not in vivo. And remember, mesenchymal stem cells do not produce bone or cartilage. They have to differentiate into chondroblasts and osteoblasts, which exhibit no blunting of immunologic response. So this is something that is often missed. If you look at systemic review articles on the biologics that contain these mesenchymal stem cells, you'll see that there's in every instance where I found no conflict of interest, they basically said there's no compelling evidence that CBMs are more likely to produce bone than just plain demineralized bone matrix and or allogenic lyophilized cancellous bone, which is what they're usually mixed with. And in a systematic review article published the in Spine, the quotation is, currently there's no available evidence to support the use of mesenchymal stem cells, bone marrow aspirates combined with synthetic or allograft materials as a substitute or supplement graft to autologous bone graft. And then in another systematic review article, although CBMs appear to be safe for use in bone graft substitutes, their efficacy in spinal fusion surgery remains highly inconclusive. With currently available data, there's insufficient evidence to support the use of CBMs as bone graft substitutes in spinal fusion surgery. Now, of course, this is not, foot and ankle surgery, but I think the principles remain the same. It's a very expensive group of products that don't really have evidence to justify their cost. Now, there are complications associated with autologous bone grafting, and those are primarily limited to the donor site. I mean, non-unions can occur with any form of grafting, but we'll exclude that for the moment. But these donor site complications are really historical in nature. If you look at the various promotional literature by a variety of companies producing orthobiologics, they will frequently cite old literature that involves open procedures in which there's muscle stripping and there's as high as 20 to 30% long-term serious complication rates. Well, this just doesn't happen with MIS techniques. And the reason is we don't do any muscle stripping. We don't do any tendon removal. We make small cortical windows, and through those small cortical windows, we harvest the cancellous bone. And this generally is a percutaneous procedure. Now, I'm going to try to convince you that not all MIS autologous harvesting is equivalent. Ideally, an MIS autologous bone harvester should avoid accidental or excessive cortical bone violation. It should utilize some form of very controlled cortical penetration. It should also utilize a harvesting means that provides for proprioceptive feedback to reduce the risk of unintended cortical breaching. And I will contend that manual rather than powered cancellous bone harvesting provides this. It should provide for a multiple redirected passes through a single cortical window. It should also have a cancellous capture mechanism that minimizes distortion of the native architecture and preserves the cellular content in its native location. This means you should avoid unnecessary morcellation and or suction that dramatically alter the graft. You should avoid cancellous bone desiccation and or minimize handling. And it should employ a procedurally efficient single patient dedicated device without need for assembly, disassembly, or supplemental instrumentation. Now, this idea that morcellation or morcellizing the bone graft and impacting it is a good idea does not have, let's just say, experimental justification. If you look at a couple of articles that were specifically intended to address this issue, one in Acta Orthopedic Scandinavia and another in Clinical Orthopedics and Related Research, when they impacted cancellous allograft into the chambers of tibia of rats, they found that the impaction of morcellized bone, as opposed to using just intact cancellous allogenic bone, resulted in unfavorable results. That is, reduced osteoconductive properties in the bone graft. It reduced or prolonged periods of time for integration and revascularization. So morcellizing and impacting does not, at least in experimental evidence, help your bone graft material. Bone grafting principles should be observed whenever possible. You want a well vascularized bleeding bone surface, at your host site. You want to debride fibrocartilaginous and devitalized tissue from the host bone graft contact surfaces. You also want to transfer graft in an optimized form. That's to avoid unnecessarily cancellous bone morcellation, desiccation, or cellular alteration. And you want to optimize the graft host contact surface. And of course, minimize donor site morbidity. As musculoskeletal surgeons, we often regard ourselves as carpenters of the body, but this characterization fails to recognize that we're moving vital structures, that's living structures. And this is much more important often than people recognize. Optimizing the blood supply at the recipient bone site, which is mentioned previously, is important. But as important, or perhaps even more important in this particular discussion, since we're talking about alternatives to osteobiologics, is the preservation of the cell vitality and the microenvironment of the bone graft. And allografts and bone graft substitutes, which are devitalized, they don't have this distinct advantage that autogenous cancellous bone graft can have if you preserve the osteoproliferative cells within their native microenvironment. If you disturb that microenvironment, well, then perhaps it's better to just revert to allografts, because you're not necessarily benefiting from the osteoprogenitor cells that are in their native microenvironment. So while carpentry is important, cultivation should not be minimized. And these two illustrations of transplanting something, in this case, two plants, one where you preserve the roots and the other where you do not, should help you understand, for illustration purposes, that we're not carpenters alone. We're gardeners. We're cultivating bone at the site, and we need to transfer the bone in as viable a form as possible. So again, avoid unnecessary morcellation, avoid desiccation, avoid handling, and avoid excessive impaction. So I think any autograft harvester that is MIS that unnecessarily morcellizes or displaces the cells from their normal microenvironment is counterproductive. So why consider the COREX bone graft harvester? Number one, it's a single patient use sterilely packaged device. It's an all-in-one harvester. There's no need for supplemental instrumentation. Number two, no assembly or disassembly required. That's a time and cost saver. You can just extrude the intact cancellous bone, which has been minimally manipulated, without disassembly. It's a manual deployment device, and I can't emphasize this enough, that the proprioceptive feedback in a minimally invasive surgery is very important. It's that feeling in your hand, as well as the auditory feeling, or the auditory perception of the cancellous bone being harvested, that helps increase the safety. And cortical breaching through our device occurs with a patented integral component that limits the depth of penetration. Also, there are castellations at the tip of our harvesting trephine and a bevel projecting surface to microfracture the perimeter of the cylindrically captured cancellous bone. We have a patented capture mechanism that allows for securing the cancellous bone without distorting the architecture or displacing the cellular elements. And perhaps the proof in the pudding is this: we have over 35,000 clinical uses since 2007 without a single serious reported complication. That is, there's no MAUD reports of a serious or life-threatening complication with the use of our device. So I hope you'll consider using, or at least looking at, the COREX device. I can tell you from having used it myself personally, that with experience, you become more and more competent at using the device, and I think you'll find it to be a very useful tool. Thank you very much.